Hello everyone, welcome to our fourth lecture video here in Unit 5 on cellular respiration. In this lecture video, we're going to discuss the process of full-blown aerobic respiration. So what does that mean? So that means at this point now, we're going to make the assumption that we have enough oxygen. So I have that right here in my laser pointer. As well as there is mitochondria present, so we're in a eukaryotic cell. So that now after glycolysis, the pyruvate here, as well as the NADH molecules produced in glycolysis, could enter the mitochondria and undergo the remaining steps of full-blown aerobic cellular respiration, which is going to be the citric acid cycle, also called the Krebs cycle, and finally an electron transport chain that is going to allow us to produce a ton of ATP energy through a process called oxidative phosphorylation, which should sound familiar because it's very similar to what we learned during the light reactions of the photophosphorylation process. So what I want you to do is there's going to be a large amount of material in this lecture video. Feel free to pause it at any point to write down or rewind and try to clarify anything I discuss over the next several minutes. So what we're going to do now is we're going to again follow pyruvate as it enters the mitochondria and gets to the Krebs cycle or citric acid cycle. But before we do that, I want to go over just a few components of the mitochondria itself. Remember, from our cell unit, mitochondria, just like chloroplast, are prehistoric bacteria that we have formed a symbiotic relationship with. They have their own DNA, so they control their own rate of division and growth, as well as their own ribosomes, in which they use to make their own proteins. For our purposes, we want to focus on these three components here, because this is where the remainder of the steps for respiration are going to occur. First, we have what's called the cristae, or the inner membrane. So, where my laser pointer is, this is the inner membrane. All right, there's the outer membrane, and then when you come into the mitochondria, you'll find another lipid bilayer. It's folded. As you can see, it dips in, comes back out, dips in, comes back out. All these foldings of the inner membrane are also referred to as the cristae. I'm going to stick with that phrase than simply saying the inner membrane. So the inner membrane is folded into this cristae formation, and the reason why it's folded, just like the thylakoids were stacked in the chloroplast, it's to increase the overall surface area that we have within the mitochondria to produce ATP. So if we just had one inner membrane that was a big round oval circle, that is not as much surface area as if you have all these little foldings in and out like you see. The final stage of aerobic respiration, the ETC, will occur here along the inner membrane, and we'll get to that in our last lecture video. For our purposes today, the next couple steps that we're going to cover is going to be within the matrix, which is the liquid-filled center of the mitochondria. So in this diagram, the green area, so the matrix right here is being pointing to nothing. This is the matrix. All right, it's just a liquid-filled area that has lots and lots of different types of enzymes that are going to be used to form various redox reactions. So what we're going to learn about today is a prep step. So something that happens in preparation of then the Krebs cycle, they will both occur within this green area called the matrix. And then finally, we have what's called the intermembrane space, which right here is just the little empty space between both the outer and inner membrane / cristae. The chloroplast had a region just like this, but it did not have any function. But now, we're going to see later on, once again in the final stage, the ETC, or electron transport chain, that this is where the hydrogen ion concentration is going to be built up that we'll then later use to make ATP. So, where do we go after glycolysis? The two pyruvates will enter the matrix of the mitochondria. And before the Krebs cycle can officially begin, we have to do what's called a prep step, sometimes also known as pyruvate conversion. So what happens is this. Each pyruvate, which remember is a 3-carbon molecule, is converted into another molecule called acetyl-CoA. So this is the molecule that can actually enter the Krebs cycle. Pyruvate is not recognized by the enzymes that begin the Krebs cycle, so it's converted into this, which is recognized. To do that, you have to cut off a carbon off of each pyruvate. So that means you're going to release a CO2 per each pyruvate conversion. All right, you're going to have to do it twice because you do have 2 pyruvates that came from glycolysis. So you produce now 2 CO2s. So now we start to actually destroy glucose to a degree. We first just split it in half into 2 pyruvates eventually. But now we're going to start to release some of those 6 carbons from glucose as carbon dioxides that we exhale. And also, finally, we make 1 NADH or electron carrier per pyruvate conversion. So overall, the 2 pyruvates here that came from glycolysis, they will diffuse from the cytoplasm into the matrix. Each of the 2 pyruvates will have a carbon dioxide cut off of them, leaving you with a 2-carbon molecule that becomes acetyl-CoA, as well as you get the added benefit of having 2 NADHs produced, which are electron carriers that we will use in the final stage, once again called the ETC. So if we look at a quick picture of this, so we're in the matrix right now, the center of the mitochondria. Here's a pyruvate, 3 carbons. We're going to cut or break this bond right here, and it's called a decarboxylation reaction. That releases a carbon dioxide, as well as it'll add a pair of electrons, as well as a hydrogen ion to NAD+ to create NADH. The leftover 2-carbon molecule will attach to an enzyme called coenzyme A. When you add that to the leftover 2-carbon fragment, that's when you get what's called acetyl-CoA. CoA is actually the enzyme that is going to begin the Krebs cycle. Think of it as a limo driver that's going to take you to your final destination. CoA is going to take this 2-carbon leftover fragment to its final destination, which will be the Krebs cycle. So again, the prep step is going to happen twice, once for each pyruvate. We'll make 2 NADHs, 2 acetyl-CoAs, and produce 2 CO2s as a waste product. Now we're ready for the Krebs cycle to begin, which is also called the citric acid cycle, because citric acid, or citrate sometimes it's called, is the first stable intermediate that's made during this reaction. It's going to happen twice because we have 2 acetyl-CoAs from the prep step. It will also occur within the mitochondrial matrix. And by the time the Krebs cycle is finished, it'll have to turn 2 times. All right, glucose will have been officially destroyed and broken down and fully released as carbon dioxide. So there is nothing left of glucose after the Krebs cycle spins twice to break down those 2 acetyl-CoAs that came from the prep step. We will produce 2 electron carriers this time around during the Krebs cycle. Again, the first one we're familiar with will turn NAD+, which is the oxidized form, into NADH, which is the reduced form. Again, carrying a pair of electrons and a hydrogen atom. And we'll have a new one, since we're looking at a new organelle, right? The mitochondria. We'll have what's called FAD, that's the oxidized version, which will accept a pair of electrons and actually 2 hydrogen atoms and become the reduced form of FADH2. So, the way I've broken it down is like this. Acetyl-CoA, again, which is a 2-carbon molecule. The CoA enzyme will add this 2-carbon molecule, which by itself is called acetate, officially into the cycle. CoA adds this 2-carbon acetate to the actual starting molecule of the Krebs cycle, which is called oxaloacetate, which is a 4-carbon acid. So this is what we begin with, 1 4-carbon acid, oxaloacetate. That's what we're going to have to end with too. So CoA adds the 2-carbon acetate molecule here to oxaloacetate, again a 4-carbon molecule. And our first stable intermediate that we make is called citrate or citric acid. All right, so that's why it's also called the citric acid cycle, although I prefer to call it the Krebs cycle after Hans Krebs, who you saw on the previous slide, who won a Nobel Prize for figuring out this process. So 2 carbons came in. That means only 2 carbons can leave. We're going to get rid of these 2 carbons. So first, citrate here is going to go through several reactions. First thing that happens is it gets converted into what's called ketoglutarate, which is a 5-carbon acid. So we remove one of these carbons right here. All right, leaving you with ketoglutarate, which only has 5 carbons. So right here is that reference point. What you'll do next is cut off the 5th carbon, leaving you with a 4-carbon molecule that needs to get regenerated back into what we started with, which was oxaloacetate. So from 2 turns of the Krebs cycle, again, it has to happen twice because we have 2 acetyl-CoAs coming from the prep step, or pyruvate conversion. This is what we're going to walk away with. We're going to walk away with 4 CO2s total, so that's from 2 turns. So that means 2 CO2s are made per turn, which should make sense because we're going to get rid of 2 carbons per turn from the acetyl-CoA/acetate. 6 total NADHs, so 3 per turn. 2 FADH2s, 1 per turn. And only 2 ATPs, 1 per turn. And this would have been made, these ATPs would have been made similar to what we saw in glycolysis through a process called substrate level phosphorylation, where an enzyme just makes 1 little ATP at a time. Very slow, very inefficient. So let's take a look at this diagram here. So where my laser pointer is, we have the prep step. Each pyruvate or pyruvic acid, you can use those words interchangeably, gets turned into an acetyl-CoA. You can see you make a CO2 and an NADH from each conversion. But let's focus on the acetyl-CoA here now going through the Krebs cycle. CoA will add this 2-carbon molecule by itself called acetate to our starting molecule, oxaloacetate. We get citrate, a 6-carbon molecule. We do a decarboxylation reaction, remove 1 CO2, get an NADH. We're left with ketoglutarate, 5-carbon molecule. Cut off that 5th carbon, release it as a CO2, get another NADH. So now the 2 carbons that came in up here have been removed as CO2. What's left over, a 4-carbon molecule that we're not going to name here, has to now get regenerated back into the 4-carbon oxaloacetate. And along the way, you can see where the ATP and FADH2 and actually a 3rd NADH are being produced. So this whole process here, again, has to happen twice, right? 1 for each of the 2 acetyl-CoAs that were made. Again, we're in the mitochondria matrix right now, where both the prep step and the Krebs cycle occur. So from 2 turns of the Krebs, we're going to get what? 1, 2, 3. Multiply that by 2, we're going to get 6 NADHs, which will go to the ETC. 1 FADH per turn, so we're going to get 2 FADHs from 2 turns. 1 ATP per turn, so we walk away with 2 measly ATPs after the Krebs cycle. And after this point, again, glucose has been officially destroyed completely. You would have released 4 CO2s total from 2 turns of the Krebs, and 2 CO2 from 2 prep steps, and those are the 6 carbon dioxides that came from glucose initially. So in conclusion, after the Krebs cycle, glucose, right here, the 6-carbon molecule has been completely broken down. 2 CO2s from 2 turns of the prep step, 4 CO2s from 2 turns of the Krebs. If I jump down here, after I bring all this information up, we only walk away with 4 net ATP so far. 2 net from glycolysis. Remember, you have to subtract the cost of glycolysis, which is why it's only 2. And only 2 ATP here from the Krebs cycle. So we only got 4 ATP so far. Well, we're supposed to get a total of 38 with aerobic respiration. So we're going to get the remaining 34 ATP in the electron transport chain by using the 12 electron carriers that have been produced as we've been breaking glucose down through glycolysis, the prep step, and the Krebs cycle. And you can see here where all these electron carriers were produced along the way.